Hello ma'am, myself Riya Surve from FY Biotech and my topic name is Importance of Sugar in Microbial Growth. So here's my first slide starts with Introduction of Sugar in Microbial Growth. Microbial growth refers to the increase in the number of microorganisms in a population rather than increase in the size of individual cell. Second, it occurs through a process of cell division, typically binary fission in bacteria, budding in yeast, or other forms of replication. The term commonly used in microbiology to describe how microorganisms under specific environment conditions such as in nutrient-rich medium at optimum temperatures with adequate moisture and pH levels. So here's my third slide of types of sugar utilized by microorganisms. First is monosaccharides, that is simple sugar. Monosaccharides are simple sugar in which there are one oxygen atom and two hydrogen atom for each carbon atom present in the molecule. They exist in straight chains or in the ring or cyclic form. Monosaccharides are reducing sugars. Examples. First is glucose, a primary energy source for many microbes. Second, fructose, commonly metabolized by bacteria and fungi. Second, disaccharides. Disaccharides are made up of two monosaccharides joined together by a condensation reaction. The condensation reaction is the joining of two molecules with the formation of a new chemical bond in a water molecule is released when the bond is formed. Examples. First, lactose, utilized by E. coli. Second, sucrose, commonly metabolized by yeast and some bacteria. Third, polysaccharides, that is complex sugar. Polysaccharides are polymers formed by combining many monosaccharides molecules, that is more than two, by condensation reaction. Polysaccharides do not taste sweet because their molecules are so numerous. The majority of polysaccharides do not dissolve in the water. Examples: starch hydrolyzed by amylase producing microbe bacillus. Second, cellulose degraded by cellulitic bacteria and fungi. So here's my fourth slide of pathway of sugar utilization. Fermentation by sugar is a biological process where microorganisms such as yeast or bacteria convert sugar like glucose, fructose or sucrose into energy and byproducts such as alcohol, acids and gases under anaerobic conditions, that is in the absence of oxygen. During this process, the sugar is first broken down through glycolysis into pyruvate, which is then further metabolized depending on the type of microorganism and fermenting pathway. In alcoholic fermentation, yeast converts sugar into ethanol and carbon dioxide, which is used in brewing, wine making, and baking. In lactic acid fermentation, bacteria convert sugar into lactic acid, contributing to the production of yogurt, sourdough, and fermented vegetables. The process is influenced by factors like sugar type, temperature, pH, and microorganisms involved. So, here's the diagram of glycolysis by ATP production. Here's the fifth slide of benefits of sugar in microbes. Sugar is a vital energy source for many microbes, playing an essential role in their growth, survival, as well as metabolic functions. So here are the key benefits of sugar for microbes. First is energy source. Microbes metabolize sugar, that is glucose, sucrose, lactose, to generate energy through pathways such as glycolysis, fermentation, and respiration. Energy in the form of ATP is crucial for the cellular process, including production and movement. Second is carbon source. Sugar serves as primary carbon source for building cellular components such as proteins, nucleic acids, and lipids. This is critical for cell growth and maintenance. Third, facilitates fermentation. Many microbes such as yeast, lactic acid, bacteria fermented sugar to produce byproducts like ethanol, lactic acid, carbon dioxide, etc. These fermentation products are beneficial for industrial applications including brewing, baking, and dairy processing. So here are some industrial importance of sugar. First, in alcohol production, ethanol is Saccharomyces cerevisiae ferments glucose or sucrose to produce ethanol used in beverages, biofuels, and pharmaceuticals. Beer and wine, sugar from malt, that is maltose, or grape juice, that is fructose, and glucose, are fermented to produce alcoholic beverages. Second, dairy industry. Lactic acid fermentation. Lactic acid bacteria, that is lactobacillus, ferments lactose in milk to product yogurt, cheese, and sour cream. Probiotics. Sugar supports the growth of beneficial microbes in dairy-based probiotic products. Third, organic acids. Lactic acid, produced from glucose by Lactobacillus species, used in food preservation and biodegradable plastics. Citric acid, produced by Aspergillus niger, using sucrose for preservation of food, beverages, and pharmaceuticals. So I would like to conclude my presentation. Thank you, ma'am, for your valuable time and attention.